Karl, you're up. Okay.
Okay. Can the student lightning talk presenters come on up? All right, let me stop sharing this, and Adrian, you're going to be first, so I'll get your PowerPoint up. And it's that one, right?
Yes.
Okay, just one sec. Okay, while that's loading, over the summer, as we were planning this meeting, we issued a call for poster presenters and student lightning talks, and are happy to present this group of students from universities throughout the Mid-Atlantic, all of whom have some exciting research to share on conservation topics. I'll let you know that one person dropped late, so your lightning talks can be a little less lightning-y. You've each got a minute or two of extra time, so take your time in getting through it. And just a reminder to the audience that following the summary remarks by Tony after this, there will be a poster presenter session upstairs in the Versailles and Pompeii rooms, which are just up there. You'll see to the right when you get up the stairs and come out. So we are going to start with Adrian. Come on up. I'm sorry, I gotta now share the screen again. 1 second. That should do it. And getting out of that. Now you're good. So you can use the arrow or that. Thank you.
Hi, let's talk about science. So in the background here is a picture, it's a snapshot of the Hudson Canyon about 100 miles off the coast of New York City. And I use specifically the word snapshot because I think that word very much categorizes the types of methodologies we use when we try to sample places like this. They are discrete in time and oftentimes don't necessarily represent the full amount of data that is available to us. We can only go out to see on very discrete portions of our time and organize, whereas these places are always existing as we are now. So my field of study tries to sort of bridge that gap in data using the various different accreting structures of marine organisms like mollusk shells, coral skeletons, and fish otoliths. And what makes these things special is that they grow over time. And as they grow, they incorporate different trace elements like barium, strontium, and magnesium into their structures through various sort of environmental and physiological processes. And this growth isn't linear. It varies throughout the season. So we get these clear temporal rings, like tree rings, that show growth over time, and that is the temporal signal that we can then work with. In the case of my master's, I work with this technique called LAICPMS, or laser ablation inductively coupled plasma mass spectrometry. Working with this deep sea fish, Pacific grenadier, off the coast of California. As you can see, this is a cross section of one of the otoliths. So looking at the core, that would denote the year of birth. And if we were to draw out a laser transect following growth margins all the way out, we would then find the year of capture. And through that, we can develop this sort of trace element concentration data across that whole temporal gradient, thereby being able to track the sort of differences and shifts in environment and migration across its life history. And this doesn't just apply to scleroside structures. For example, take a look at this flounder, look at that face. If we were to rip out its eyes and slice them open, we would see the same accretionary bands. These are not annual, but they are inert and they are proteinaceous, meaning that we could sample the stable isotope ratios of across time and then sort of generate an idea of the dietary shifts and trophic position across ontogeny. And in the deep sea, we can even do things like trace sources of chemoautotrophic productivity and map food webs, etc. One of the panelists talked about limitations. I'm going to talk about limitations. I'm currently working with this cold water coral species, Desmophyllum dianthus. And one thing about my field of study is that we might not necessarily know the exact sort of biomolecular mechanisms for how these structures form. It's still sort of a black box. And in that way, we are just interpreting the end products of a far more complex system than we really understand. In addition, with these samples, they are very complex morphologically, and I've had to do a lot of work with fancy techniques like micro CT scanning to be able to develop an idea of how these structures are actually forming over time. And developing more advanced laser techniques like 2D elemental mapping, looking at the trace element distribution across the structure itself. So why is this sort of data important? One, I think it's far underutilized in our field. In addition, in places like the Hudson Canyon, which operate off these very long time scales, it's important that we get this long-term data sets. And we keep talking about the word baseline and how we need to incorporate an idea of what that baseline is. And the thing is, for places like these, we'd already changed the planet quite a lot before we even knew these existed. So to be able to develop a baseline of these systems prior to perhaps even us knowing about them in the first place, especially for cold water corals that have existed for, like, they can live for maybe 300 years. The deep sea fish I was working on was like 70 years. These are very long time spans of data that we are completely ignoring. And it's one way we can sort of glean more data through less effort. So thank you, and I'd love to take any questions.
I think we'll hold all questions to the end so everybody can get them at once. And next up, Jake. Just give me a second while I switch up to your presentation, which is hopefully loaded on here after this one. Is that you?
Yep.
Perfect. All right. There you go. Thank you.
This is a pointer.
There is a pointer if you hold the top.
No, not really. Okay. It wasn't showing. Yeah. All right. Well, hi. My name is Jake Kuenzli. I'm a second-year PhD student at Rutgers University. And today I'm going to give you a quick overview on basically my first chapter, which is looking at species-specific trends in about 30 years' worth of New Jersey DEP bottom trawl data. This work was done in collaboration with Drs. Jason Adolph and Keith Dutton here at Monmouth University, as well as my advisor, Grace Saba at Rutgers. And so this talk is really going to build on some of the themes that we've kind of heard throughout this meeting, both today and yesterday. So we've heard a lot about ocean warming here today at the meeting. We've also heard a little bit about shifting distribution. So I'm not going to go too far in depth as they did, but so basically ocean warming is happening, and it's happening at a much faster and accelerated rate in the northeastern United States. And as a result of this temperature increase and the associated physiological and behavioral effects, we're starting to see these changing distributions for marine species as they're moving further poleward and they're shifting further down in depth. Now, none of this would be possible without New Jersey DEP, so I am going to have to shout them out because the ability to have a data set like this going back 30 plus years to be able to look at fish abundances and species abundances is very valuable. So big shout out there. So if you're not familiar, the New Jersey DEP bottom trawl survey is done to cover all 4 seasons. It goes out every year. Sites are selected randomly, and they cover both nearshore to offshore environments and north-south from the mouth of Delaware Bay to Sandy Hook Bay. So the study aims, when we had this huge data set, and I mean huge data set, I put it into R and it broke, was to look at the most abundant species over time, look at presence and absence, so basically to see as their abundances change when they kind of dropped off or started coming in, and then of course those species that really popped up kind of out of nowhere after not really being in the trawl in previous years. So to get into some results, I'm going to walk you through this graph here. So how this started was going through the 30 years, breaking into 10 decade spans, and looking at the most abundant species by catch for each decade. And by going through those 3 decade spans, picking out the species that only appeared in the top 90 twice or once. So on this left side of the graph here, where it's a count of 2, that means that these are species that are present in 2 different decades, some of them simultaneously and some of them have gaps in the middle. And then on the right side, they were only present in 1 decade. And so where your eyes might go to first is these ones here in orange, and these are the species that only recently came into the top 90 in the most recent decade. And now I do want to say this doesn't necessarily mean that they weren't present during the other 2 decades, but it does mean that they had an increase in their numbers enough to break into that top 90 when they weren't originally able to do so. Now, the other group that I would want to draw your attention to are these ones here in purple, because they really represent a really interesting thing that's happening in the environment. You have ones that were present for the first decades, that's 88 to 98, but then they dropped out of the top 90 in that middle span, and then came back in the most recent one. And so while you might be able to start thinking about the ones that recently appeared of being like an environmental change, like the accelerated ocean warming, it does pose a different question for those species of what drove that shift in abundance. The other thing that I looked at was the change in catch over time for elasmobranchs, because with such a huge data set, kind of had to boil it down in certain ways, so I wanted to look at a group of organisms to see how they were changing. Also, from talking with recreational fishermen, there's been a lot of evidence of shark species increasing in abundance in our region recently. So we wanted to look at elasmobranchs and see if the trawl data could kind of corroborate that. And you will see that outside of a few species, namely some of the skate species and smooth dogfish, that largely most of them have experienced this positive trajectory and a substantial increase in the most recent years. And now, since I do have a little more time and a little less lightning, I do want to mention that this has been kind of a jumping off point for the next 2 chapters, looking at some of these species and actually focusing on their distributions and habitat use, as well as some vulnerable shark species not featured on this graph, to kind of get at that whole question of how is the elasmobranch distribution and habitat use changing in a world of ocean warming and environmental change. Thank you.
All right, our final speaker, Diederik, you have no slides, right?
I did. I sent them to you. I also have them in the phone drive.
What's that?
I sent them to you this morning.
Oh, this morning.
Yeah.
Okay, do you have a flash drive by any chance? Cool, let's do that. Yeah, while we're loading up those slides, any questions for the first 2 speakers? Take a couple. All right, we are almost there.
There we go. And I apologize if I forgot both your names, my bad. But for the first speaker, you were having some samples from those deep sea canyons. how did you come to have those samples? Just curiously.
I may have not mentioned, these samples are not from the deep sea. They are, Desmophyllum dianthus is cosmopolitan. They're found all over the world. I think if you were to talk to some ecologists, they'd argue that there's a good chance they're not the same species, but at the same genera, ideally they're probably incorporating, they're still growing at the same sort of mechanisms. This species I sampled from a Patagonian fjord in Chile because fjord ecosystems are very unique and they allow a lot of deep sea animals to live in them. So that's one place where they're shallow enough at like 7 meters to be able to actually go down and grab them with some scuba gear. So no, I wasn't on like a cruise or anything like that.
You didn't get to go to Patagonia?
I did go there.
Oh, you did?
Actually.
Okay.
Oh, and one thing I can mention about is that with these specific samples, you can work with anything that's preserved. I was working with historic samples for my master's. These were captured in the 1970s. So like we have plenty of museums chock full of these specimens that we can still rifle through and look at retroactively. We don't necessarily even have to go out to sample them. We already still have those data sets. We just have to actually open up some drawers and stuff to look at them. So.
All right, thank you. We're all set now with Diederik. Go ahead and introduce yourself.
Hello, everyone. Sorry about that. My name is Diederik Boonman. I'm an undergrad student here at Monmouth University. And recently, me, with the help of my 3 co-authors, Drs. Craig Walding, James Carlton, and Jason Adolph, present in this room, we recorded or published the first scientific observation of Actinia equina in the United States, which had never been seen anywhere in the Americas beforehand. And what it looks like is it has a pale green base, like you can see in these photos. It has retractable tentacles, which is what you see in the 2 photos on the left. It has a blue limbus, and that's the blue circle around the base of the anemone, and a gray pedal disc, which you can't see in these photos, unfortunately. The tentacles are a similar color than the base with blue tips, and the size 10 to 40 millimeters, and tentacles 92 to 143 tentacles for this species. And once we had all this morphology that we were able to see, we had no idea what it was when we saw it the first time in the at the beaches, and we had to find out what it was. So to do that, we did some genetic analysis with ITS and COI sequencing, and it matched known markers of Actinia equina, which is this species right here. And as you can see, it has high phenotypic diversity within this species. There's a lot of colors. You can find it from red, orange, pink, yellow, brown, green, presence or absence of the blue limbus that I mentioned before. And because of this high diversity of phenotypes, it was at first a little bit harder to identify just with morphology, and that's why the genetic analysis was very helpful in this. And it does match the morphology of this species. So originally, this species is found mostly in Northern Europe, as well as in more scattered in other parts of the world, and now in Asia as well. And they live in rocky intertidal zones, like this photo right here. This is a photo of San Francisco, but it's very similar to the areas where they live. And we don't really have this ecosystem in the shores of the mid-Atlantic, but instead they have invaded these human-made beach groins, or as New Jerseyans might call it, jetties, that we constructed for sand retention. And this provides structure, shelter, and food that creates a different habitat in an otherwise sandy environment. And now we are creating a map showing where they are and where they're not. We have found them in the green dots from this, which is Long Branch, pretty much where we are right now, down to Manasquan, which is about 20 kilometers south of here. The red dots, we did not find them there. They're not present in that area, or at least that we can see them. And the black dots, we were supposed to sample this past weekend, but with Hurricane Helene, it was a little bit difficult to go out to the beach. So how did they get here? The most likely vectors of introduction is from shipping from Europe, either through ballast water or whole fouling. These anemones produce clones that float in the water and can survive extreme conditions, and they just wait for a good substrate for them to be able to settle and grow. So either through clonal dispersion in the ballast tanks or whole fouling, the entire anemone coming over. And a less likely vector of introduction is the aquarium trade. This is a tank that we set up for observation, but in doing that, we found out that Actinia, the whole genus, is very well known to be very common in aquarium trade. Potential impacts, they could have invaded also artificial reefs, which, as you might know, we have a lot of them in New Jersey. We haven't been able to test that because we haven't been able to go down into the water and see them, but there could be impacts on those structure-dependent organisms like other invertebrates like you see in this image on the left, or fish that feed off of those organisms. And aside from that, the environments that we do know that they invaded, the rocky groins or jetties, it's a fairly understudied habitat and very new, meaning we don't understand that much about the dynamics that go on in there, and because it's so new, we don't have that many native species that take advantage of this environment. So it created a niche that could be taken advantage, and in fact it has been. There are two other invasive species that make use of this space, Quercinus manus, that's a European green crab, and the Asian shore crab. So now we have three invasive species in the same environment that was created by humans. So what can you do about it? You can help us find them. We are trying to get the range of where they are and where they're not. There's only so many of us that can go down, up and down New Jersey, or even other states. So you know what they look like, and where you might find them is in those rocky groins or any sort of hard structure in the intertidal zone. They tend to be around or below where mussels are. That's the height that they prefer. And yeah, if you see them, you can either report them on iNaturalist, which is a tool that we used a lot for this project, or email me or Adolf, Dr. Adolf, with the location and date of the observation. And understanding this range will help us know how to react to this recent introduction. Thanks for your attention.
Thank you. We will open this back up for questions, but I do have a first one, Diederik, for you, two actually, while we're warming it up. Can you tell me a little more about how actually you found the first one? What were you looking for when you stumbled upon it? And two, do they pose any kind of a sting hazard to bathers or anything like that that we should know about?
So that's a good question. We found it originally during a marine biology class. We would go to the rocky groins to study and learn about zonation. And actually it was Dr. Adolf's class. And we were just looking at the different organisms that grow in different parts of the intertidal zone. And we were just looking at the things and kind of knowing the type of organism it was. But I've always liked taxonomy and identifying things. And we couldn't figure out what the anemone was. Like, none of the books, nothing on the internet could tell us what it was. And so we started reaching out to anemone experts because neither of us have any experience with anemones. And that's how we slowly started creating this, the basis for this project. And I forgot,
Part two, yeah, human interactions with it, any issues with that?
Right, so they are cnidarians, so they do produce a toxin, but their tentacles and their cnidocytes are too brittle and small to penetrate human skin. So they don't really pose a big threat to humans. Aside from that, they're pretty small and they only grow in crevices. So they're not quite exposed to like where humans might be. But as far as we're aware right now, there's no threats to humans directly by touching them.
Hi. Okay, this works. Adrian, I have a question for you. I don't, and sorry if I missed it. The environmental variables that you're able to pull from the layers, are you? First of all, my first question is, what are they? If it's So if you can answer that, then I can have a second part.
Yeah. In fish, they've mostly been used to trace migrations of anadromous fish from freshwater estuarine environments to the ocean. So barium and strontium seem to be two pretty well-known elements that are often used to trace salinity. And one thing that is an issue in this field is that reproducibility is very difficult. People have a lot of different methods for how they sample or use the parameters of the laser itself. And also, there are differences between species. There's differences between different bodies of water. In the Baltic Sea, magnesium seems to be a very good indicator for hypoxia, but that hasn't been shown to occur in like the Southern California Bight or anything like that. So more research into developing stronger proxies and understanding these proxies at scale are sort of where our field needs to head.
That's great, thanks. Yes, so the second part of that question
Yes.
was, when you can see environmental variables like, a proxy for dissolved oxygen concentration, are you able to pair that with other, characteristics of the structure for any indication of like organism health or growth rates?
There's multiple different things you can do. So one of the things is like just flat growth rate. Like if a band is smaller, that means it grew less. And this actually does correlate with like total length or body weight, depending on the organism. I know for fish, yes. And that has often been used to generate sort of these long sort of biochronologies that I've tried in the past and have never gotten to work. But when it does work, you can generate this really cool interannual signal of growth per year. And this has been done this is a technique we borrowed from tree scientists, from dendrologists. And you can look at that and compare that to like interannual variations like Enzo or other ones. And that has been paired out to sort of like show that there is a global sort of trend in growth. And there's certain elements that seem to be more physiologically mediated than environmentally. Again, I think those are less understood than strontium, barium, magnesium are the big three, I'd say. But everyone, you can tell the machine to look at anything, basically. So like there's plenty others to look at. But yeah.
Is this on? In the DEP data set, have you found any, have the environmental variables that are collected with the fish data proven useful in explaining some of the either appearance, disappearance, reappearance, or trends in the fish you've been looking at? Have you looked at that yet?
It's been helpful from the standpoint of getting a temperature picture from when the trawls were conducted and when the species were caught. So obviously this is a shorter talk. I couldn't include all of my data products, which is a lot. But I do have plots for temperature, both bottom and surface, over each 4 seasons. And you can see a upward trajectory for temperature throughout the trawl from the beginning to where my data set ended. So the temperature did support increasing temperature possibly being the reason for seeing an increase in abundance for some of these species, especially in recent decades.
Yeah, I remember you showing me that. I was thinking more about I forget which fish they were. I couldn't really see it from here, but ones that were there in the beginning, and then absent, and then there again. Is there any correlate, any environmental correlate in the data that might explain that?
Looking through the data from the environmental, I have not seen anything that explains that drop off. I can speculate possibly that it could be something prey driven as opposed to more of an environmental parameter, but that's just not something that I'm sure about, and I don't have anything in the DEP environmental data set that supports that.
Thank you.
Hello. My question is for Diederik. Do you suspect that, as you mentioned, artificial reefs, do you suspect that these anemone will start populations on oil rigs or wind farms? Because we have a lot of those here.
Not that I'm aware of. They're an intertidal species, so they only go up to about 20 meters in depth. I'm not sure of the oil rigs or structures that you're mentioning, but if they are within that range and there are currents that could bring them there, then possibly. The most likely reason why they ended up where they are is from the New York harbor, the water coming out of there. There's a certain current that pushes the water down towards the Jersey Shore, and that's where it ended up here. If there are currents going from either where they are present right now or where they are present right now going towards those structures, and there could be, but it's not something we've thought about.
I have a question for Diederik, if that's okay.
Go for it. Yes.
When you kind of showed like the map of their the range where they've been found.
Mm-hmm.
Are any of those areas where they've been found another example of them being invasive, or is that all native range for them? So it's like, is the U.S. East Coast the only instance of them popping up as an invasive species?
So the whole genus has a very interesting history, where it used to be, people thought that it was very, very widely distributed, just like a cosmopolitan species. And then through the years with genetic study, they ended up finding that there's like, I think 8 or maybe more different species that are in different ranges. And we have even another species of the same genus that is in the Caribbean. But that's a different species. So it's not very clear to me, that data, if it came including those species before they were separated, or otherwise, I'm not really sure. But as far as I'm aware of this species, I don't remember seeing them being invasive somewhere else.
What a great bunch of talks. I have questions for all of you that probably should wait till the poster session or something. But when I first heard about this occurrence of a new anemone, like that's anytime a new species shows up, people pay attention. That's like, there's a story there. And so I Googled it, and I went down this rabbit hole for a little bit when I was probably supposed to be working on an expense report or something. And you probably came across the story of Granny.
Yes.
Yes. So this same anemone became a sort of a celebrity in Europe. There's a Wikipedia page about it back around in the early 1800s, and by some accounts people came to see it in an aquarium, and it was before there was such a thing as aquariums, like Mary helps run up in New York. And there's such a, since we talked a lot about storytelling and connecting with people, I just thought I'd mention that. I was curious, also really interested in in looking at shells to learn things about the world and how it works with the Desmophyllum, which is a species that also occurs, as I think you mentioned, in the Mid-Atlantic region, at least the genus. I'm not sure on the species, but there's another species of hard coral, Astrangia pocillata. It's the northern star coral, and it's a very common member of the communities around what we call hard bottom or live bottom patch communities that are sensitive, typically mobbed with sea bass and other species of interest. And I just think that there would be, if anybody's looking for more work, there would be really a lot of interest in looking at how, I don't think anybody, to my knowledge, nobody really knows much about, less than we'd want to know about those communities in general, and specifically those cold water corals that are right here off the beach, within 20 miles of the beach, at depths which wouldn't be that hard to sample, and probably are also going to be early colonists, I would speculate, of some of the wind power installations. So I guess I'm not sure if there's a question there, I guess. Have you heard about that, and do you think that's interesting?
No and yes.
All right. I think we've about reached our time here, but there is a poster session, even for you guys who aren't presenting upstairs. Plenty of food and beverages up there and chances for you to talk more with the crowd about your research. So thank you very much for coming. Let's get back on this. That might be where it ends. Okay.
Okay, thanks. This is really going to be a pretty brief wrap-up. It's been a long and really great day. So again, as Karl mentioned, two things. The poster session, and also I really recommend, particularly because Karl has been the champion of this project, you've heard a lot about the Mid-Atlantic Ocean Data Portal, but we've actually had some students at Monmouth working on developing an app, so you'll be able to access it on your phone. So really, really encourage people to do that.